So, Dr. Katcher, so your team recently published a paper on bioarchive, so reversal of biological age in multiple rat organs by young porcine plasma fraction.
Right.
Some updates to your work on E5. So could you talk a little bit about, so what is new in this latest paper? What's kind of the updates in it?
Well, as you detailed in your own podcast, I revealed the secrets of E5, which is basically two things. One is that it's primarily nanoparticles like exosomes that are the basis of rejuvenation. And that completely disagrees with what was previously thought by people like the Gun Boys and Saul Valeda, etc., Amy Wagers, who all attributed anti-aging of blood plasma to small molecules, proteins. I never believed that was true for some obvious reasons. For example, any single gene can be mutated, and the lack of that gene or the over secretion of it should produce vast differences in lifespan, and that doesn't happen except for lamin A, and even that's a question of alternative splicing or alternate splicing. And the second thing is, and I consider this really the most important thing because it makes it a viable option for all of humanity, is that it was porcine plasma, not human plasma. And that leads to two things. One is the fact that porcine plasma is apparently, or rather the nanoparticles, the exosomes, etc., in porcine plasma don't have any immune reaction in rats or in that, for that matter, in human beings. In the one instance that I successfully tried it, which is my own hand.
Exosomes. So we think, or we know they're exosomes. So within the exosome, do we know, because exosomes contain a number of different factors, right?
Correct.
And
Correct.
so do we know which ones are producing the effect?
At this stage, nobody knows. I suspect it's microRNAs, which are significant in controlling translation.
Mm-hmm.
I suspect it's also RNA binding proteins, which are very much disturbed in the aging process, and RNA processing proteins.
So at the moment, so we have exosomes, and the exosomes. So it's really interesting that the exosomes work across species, right? So
Yes.
is it?
So this is a highly conserved process, obviously. I mean, at
Yes.
least it works across all all mammalian genera,
Right.
as far as we know.
Is E5? When you're narrowing down E5, so E5 is just exosomes. Is that it's just like pure exosomes, or are there other elements from the
We
plasma left?
we There are contaminants, of course. For instance, albumin seems to be a contaminant.
Mm-hmm.
But what I'm wondering, and what we don't know yet, and we're doing experiments now to find out, is whether there are other nanoparticles aside from exosomes. There are also what they call ectosomes, which bud from the cell surface. There are exomeres, and what they call supermeres, which are even tinier than exomeres, and both are tinier than exosomes. And uh, my purification method was designed to capture all of them.
Mm-hmm.
So, hedging my bets, as it were. Uh, so, yeah, we don't know yet, but we're doing experiments to find out.
So exosomes come out of cells. Do we actually know which cells these exosomes came from, or does it matter?
Well, I think that's the point. Uh, Exosomes come from pretty much all cells, and the exosomes in blood come from all cells.
Hmm.
And different exosomes contain different contents and have different targets. The contents are called cargoes in the literature. So exosomes have different cargoes, and they have different targets. So our thought was if we take all the exosomes, which could only be found in blood,
Hmm.
and inject them in fairly massive amounts, which can't be done with human blood,
Mm-hmm.
uh, that we would have the effects that we had. So, for instance, in the paper that you referred to by Shamagian,
Mm-hmm.
uh, they used exosomes from cardiomyocyte spheroids, and
Mm.
those seem to have a general effect. But it took 4 months of injections, which to a rat would be equivalent to about 10 years in a human, and these results occurred in 4 days. And even with our rats, we saw the effects in less than a week, or
Mm.
approximately a week. So, which even in human terms would only be a couple of months at
Mm.
most. Uh, so we think it's a combination of the variety of nanoparticles plus the amount of nanoparticles that that make all of this possible, and within a limited time period. You don't want to be
Mm.
taking injections for 10 years to see an effect.
No, no. So, I mean, could it be a dose thing? How confident are you on the dose you're using?
Um, we're pretty confident because when we have the dose, we had no effect, or a very transient effect that soon disappeared. So I think dose is a very important part of it. And
I,
this is again something that you can't do with human blood unless I hate to think of the ways that one might get such material.
yeah. I mean, there are like placental-based exosomes which you can get, but I guess it's the quantity that would be the problem.
Exactly, exactly.
So when you looked at the rats, so it rejuvenated all the places that you looked, right? Well, except for the brain. The brain was somewhat rejuvenated, but not as much.
Let me say that the hypothalamus of the
Well,
brain
all right,
was
the hypothalamus.
somewhat rejuvenated because we saw significant cognitive effects. We saw a significant improvement in cognition among the old rats.
Hmm.
Eventually, after training, they got down to the same level as the young rats, while the untreated old rats just kind of didn't improve whatsoever and actually got worse with time.
Hmm.
So we actually saw reverse aging,
Right.
youthening, I called it.
Yes. So you did run an experiment for females only, right? To try and see how long the total lifespan would be.
Correct,
Yeah,
correct.
and
That was a
that
choice of our PI
right.
in India.
And
It
and that
was
that showed
female.
that showed some improvement for the oldest. Yeah, I mean the oldest
Right?
rat was
It showed improvement for all of the rats,
mm,
but for the oldest rat, Seema, which
mm,
uh, which means boundary or limit in Hindi, uh, she lived to the equivalent of about 150 years in human terms. So a really significant difference. 6 months longer than the
mm,
normal lifespan,
right.
which again, with rats, 2.5 weeks is about 1 year of human life.
Yeah. And you stopped. I mean, it was every 45 days, right? And but then
Yeah.
you stopped at some point. Did you continue?
Yes. Quite honestly, I wasn't in India at the time, and I don't know why
No.
they stopped.
Okay.
I would have liked to have seen it continue,
Uh,
but they stopped. But even so, she just lived and lived and lived.
In the connection between nutrition and optimal hormone function, one mineral stands out. That is magnesium. It regulates hormones like insulin, thyroid hormone, estrogen, progesterone, testosterone, and cortisol. For example, a study with 399 men over 65 found that higher magnesium levels were linked to higher testosterone levels. Magnesium breakthrough from Bioptimizers is the only supplement that offers all 7 key forms of magnesium. This includes highly bioavailable forms like magnesium glycinate, malate, and orotate that raise cellular magnesium, supporting optimal hormone production and regulation. Since I started taking Magnesium Breakthrough, I feel more energized, my mood is more stable, and I've noticed improvements in other areas affected by hormone imbalances, such as sleep quality. If you want to try Magnesium Breakthrough, just go to bioptimizers.com/modern and enjoy 10% off with the code modern10. It comes with a 365-day money-back guarantee. And if you subscribe, you will ensure your monthly supply. Thank you for your support. I mean, how often do you need to renew like the stem cells?
You really don't. It's
Yeah.
they like a an embryonic stem cell divides very, very well. It's naturally occurring, it's very young, and once those things are isolated, they can last hundreds and hundreds of years of dividing. You could, in a single stem cell line, if you didn't stop it from growing, the earth would be covered in about a 12-foot slurry since they were first developed in the late 90s, or isolated, I mean. But you know,
Mm,
uh, uh, iPS cells, when you take human skin and then bombard them with a whole bunch of factors that
mm,
genetics that are expressed by young cells, kind of tricks the skin cell into thinking that it's a younger embryonic cell, and it back differentiates and becomes a young stem cell. So it's another way of making it. But the
mhm,
means of making it, jamming in 1,500 copies of 4 different things, beats up that cell. So the cell is a little less capable of dividing. And so it'll stop. It might do 10 or 20 divisions and then start to differentiate and you lose your pool. Whereas embryonic stem cells have a much more robust ability to stay young. And therefore the banking of them is really one cell has really, you know, formed the foundation of almost every treatment that my laboratories have ever generated in the last 30 years.
And actually, you're not using the cells. So you keep the cells. The cells are like a factory that are generating the secretome and you're extracting the secretome. Would that be correct?
Yeah, that's exactly right. It's very interesting. We are, in a brand new drug, in a brand new drug category. It's very fun at Immunis. The 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 cells that we start with are stem cells, and then we make these young precursors that secrete this immunological modulator soup, if you will, of 440 factors the secretome. And we just keep farming the cells, but the product is actually just the proteins, the secretome, what is secreted. So there's no self or non-self on those things. They don't get rejected. So therefore, this is off the shelf. It is something that you can just use from anybody because your immune system factors are like mine. My TGF beta is like yours, my IL-10, my IL-2. They're so similar across genders and races, ethnicities, that we can actually make them. It's called recombinant proteins synthetically and use them across genders and ethnicities. So getting them from one particular cell line is good. Basically, they're not rejected by any other individuals. So it's off the shelf. It's a secretome that has 440 active ingredients that targets the immune system in 440 different ways. And that is really the special bit of what we're doing. The immune system is so complex. It is so incredibly complex. I'm a neuroscientist. I thought the brain was complex. Started getting into immunology about 15 or 20 years ago. And my goodness, it is so, so complex. For every physiological thing that your immune system does, there's 5 or 50 things pulling and 5 or 50 things pulling. It's like an orchestra that has a beautiful, harmonious output, but it's a lot of work to get that output. And drugging that with a single active ingredient, like every drug you've ever taken in your life, every drug is a single active ingredient. But back in the days when botanicals were coming up and lots of good medicines are being made from plants, the problem that the FDA had at that time is batch to batch variability. You know, you boil a pound of willow bark and you get a different amount of aspirin derivative every time. So it's hard to make. It's sort of like a terroir in wine. So as you know, botanicals are not generally regulated by the FDA because of the batch to batch variability. Whereas cells, the developments in cell culture have been happening in leaps and bounds over the last 20 years. My laboratory at the forefront of this. And we're perhaps the world's best at growing homogeneous populations of stem cell derivates, meaning we take stem cells, make them into something in purity. Oligodendrocytes, a spinal cord cell type that's missing after spinal cord injury. You make that in purity, and it restored quite a lot of function to quadriplegics in clinical trials. We've made high purity cardiomyocytes and neurons and neuronal progenitors. And in this case, a cell type that's young and secretes every factor relevant to immune system development and health. It's an interesting new drug category, the secretomes. Just a few years old. I believe that Immunis is only the second company in the world that's making them, or in the United States. Excuse me. And 440 actives in one drug.
That is, yes, that's really interesting. So there were a couple of papers that talked about using exosomes, so young exosomes, to rejuvenate other animals. And that was actually cross species, which was a bit strange. But how much of the content of the secretome is the exosome, and how much are the other functions, the other proteins?
An exosome content of a secretome is generally 0.5%. Right. Exosomes have been a kind of a new thing in the field. They're pretty amazing. They're little bundles of genetic material that have to get out of a cell and then go across a space and get into a cell, dump their contents, and then alter the genetic expression of the recipient cell. A lot of steps in there. Very different from like a little protein that's released by a cell. You call it a growth factor. And that growth factor just simply has to, diffuse a little ways. When it hits the outside of the new cell, it hits a receptor and boom, triggers an activity. So exosomes have more steps, but they do different things. So they're both very good. Immunut contains both.
Right. So do you take the complete? Do you take the complete secretome?
Yeah, we take the whole secretome. It's it's the truth that we simply don't know what the active factor is.
Mhm.
We have a really good idea from literature, like immune modulation of muscle is done by follistatin and fetuin, and playing a big role in that. And those are two of the highest expressing components in our 440 component secretome. To knock them in, knock them out, investigate which 5 or 50 of the 440 are doing the real job is, in our view, secondary. I've had a thesis, a foundational philosophy, if you will, being a stem cell researcher in my whole career, and that is mess with the system as little as you can. Life is way better than us. And, when you try to simulate the brilliance and artistry and mastery of the human body, you fail. There's no scientist that can keep cells alive forever like the human body can. There's nothing like this vessel of ours. So as a general thesis, mess with it as little as you can. These young cells that modulate our immune system are already doing their job, and how they were programmed to do so, how their epigenetics is altered. They're doing a really good job of it on their own. Let's just capture them and let them do the job. So we are working with the right defined cell population that makes 440 factors that are identical to what you have in your system. So we've got a system here that's already doing the job for us. We just had to do the high purity derivation bit where we feed stem cells, push them along a developmental path. They want to become every single cell type in your body. It's very hard to control them, but we've gotten good at it over the years, and we are the leading group in the world doing this. And in this case, we made the right population for modulation of the immune system.
Right. That sounds like a great way of looking at it, I must admit, because it is just so complex. Like just tweaking one little thing at a time just doesn't work.
Yeah, like think about if you had to think of what 440 factors need to be secreted together to modulate your immune system. I can tell you for certain that we as a community, all immunologists together, the entirety of immunological knowledge on the planet does not know that. We don't know. It's so in this case, we're letting biology do it on its own. So what I did is I reached out with my team to 7 of the leading researchers in the world, each one of them on a different manifestation of immunological decline. So one professor, one of the leading scientists in the United States on muscle atrophy, another one on arterial stiffness, another one on adaptive immune response, wound healing, neuroinflammation, metabolism, etc. All of the major manifestations of aging, all of which we know are immune mediated. Because we have a multi-active immune modulator, I wanted to test it out on every animal model of human age-related and immune-related decline. And in all cases, we saw phenomenal benefit. We ended up grabbing onto muscle atrophy and metabolism for a couple of good reasons. One is that there's no competition. There is no approved way of changing muscle mass and gaining muscle mass. There's no drug that does that. Metabolism, we've just seen some pretty remarkable metabolic modulators recently with the GLP-1s and Ozempic, shrinking fat, but they're also shrinking muscle. That's very bad. So, we honed in on, huge market, but also driven first and foremost by dire medical need, and started working on rodents with muscle atrophy in a big way. So these are aged animals and also immobilized animals. Think about having a cast, you lose your muscle. So either immobilization or age are two things that for certain, without a doubt, no escaping it, you get muscle atrophy. So in these models, when given our drug Immuna, either the immobilized or the aged immobilized, they actually grew muscle and they got leaner and stronger. Their whole body coordination was better, right down to the cellular work of myotube fusion and increasing by 2 or 3 times the amount of young satellite cells. These are the cells that have to grow to build muscle. You take Immuna and you get more of it, even right down to the genetic expression patterns that change in order to do that. So right from, it's soup to nuts. It's right from the genes to the cells to the size of the muscle and strength of the muscle and balance and coordination. And in aged animals, Immuna certainly reversed metabolic and muscle atrophy. So I just ran over muscle. The metabolic was just also fun to see. Professor Drummond has these very expensive metabolic cages, and you put mice in them and they measure everything. Well, the amount of activity of these mice is about doubled. So for some reason, the animals are just moving a whole lot more day and night, double. They're consuming twice as much oxygen, excreting twice as much CO2. They're eating more, they're defecating and urinating more. When you look at their fat, it's reducing in size as well as cellular. The number of cells in the fat is decreasing as well as the whole fat. Pads are all decreasing. Fatty liver decreases. The toxic lipids of one's metabolism, these are biomarkers. As we age, our metabolism gets poorer, and we get a lot more toxic lipids in our bloods. These are things called ceramides or triglycerides or diglycerides. And as you're older, you have more of them. In the aged animals, they dropped down to about half when they received Immuna. All of this metabolic benefit in the very same animals, they're also gaining lean muscle and having all those muscle benefits, balance and coordination that I mentioned. So we were extremely enthused. These two works are published in very, very good journals. Aging Cell just accepted and is in print for the metabolic work. And Geroscience published just late last year our muscle work. So very good journals, impact factors of 8. About 95% of journals have an impact factor of 1 to 3. So these are very good work. And then we applied to the United States FDA and we were awarded a phase 1/2A, 9 outcome measures of efficacy and about a dozen outcome measures of safety. This is a first in human test, and it's been going on over the last year at the University of California Irvine Medical Center, UCI Health. And I'm just absolutely delighted to say that in older humans that are immobilized, so they can't exercise. This is immobilized elderly humans. We're seeing their muscles grow. We're seeing them get stronger. The muscles get stronger and the whole limb getting stronger. We're seeing in a functional quality of life measurement recognized by the FDA and CDC as a quality of life measurement. It's called the 6-minute walk test, that they are statistically significantly improved, 20% faster and further walking within 6 minutes. Their pain is dropped 50 to 70%. Their immune systems, the chronic state of inflammation of their immune systems is dropped by about half. That's extraordinary. 3 months after treatment, these 60 and 70-year-old people have an immune system that is characteristic of someone decades younger than them. Their basal state of inflammation is dropped dramatically. So it seems to be working and no side effects, which is wonderful. So this is not published, the human work yet, because that just rolled out over the last month. So we're collecting all of the rest of the phase 1/2A data through to August of this year, when it'll completely end. And we have prepared a phase 2 already because the data are without any side effects, no adverse events, and we're having tremendous efficacy. We have already gone ahead and written the entirety of the phase 2 clinical trial, in which we're going to test both muscle and metabolism now in a much broader range of humans that have a broad spectrum of metabolic and muscle disorders. This is called a basket adaptive clinical trial that has a lot of people in the beginning, and it's expensive because there's a lot of people, but it's de-risked because there's a lot of people, and you can adapt it to the responders in order to ensure that you're really focusing on what works best and giving humans that. but you're also taking a broad look at many outcome measures in order to try to capture as many benefits as we can.
Was that you are studying exosomes in your lab. So could you talk a little bit about what exosomes are and what you're studying them for?
Yeah, great question. So exosomes are a type of particle, and there's a number of things that are released from cells that are particles. They're called extracellular vesicles. It was thought that cells release these vesicles that have DNA, protein, RNA, and lipids, metabolites, as kind of a way of getting rid of the garbage. They were called the garbage can of cells. But it turns out they're not a garbage can. They're actually ways cells communicate. So they release these vesicles carrying signaling molecules, and they'll be taken up by cells at a distance. And so this regulates the immune response, it regulates metabolism, it probably contributes to everything that the body does. And it appears also that senescent cells release certain types of vesicles that have different contents than a non-senescent cell. But where we started to look at EVs and aging is the fact we observed that stem cells, so if you have young functional adult stem cells in your body, they release vesicles that function to suppress senescence. So if you add them to senescent cells in culture, they suppress the inflammation, all the inflammatory factors those senescent cells are releasing. So we have now been trying to figure out what components of these vesicles contribute to suppressing senescence. And then how does that suppression of senescence, what's its role during normal human aging? So if you have endogenous stem cells that are releasing these vesicles that suppress senescence, when and how and why are they doing this? We are trying to also use them therapeutically. So if you make enough vesicles from young functional stem cells, that you can then inject them into a mouse and eventually a human, and they should suppress inflammation and senescence. So they could be used therapeutically. And I guess the other interesting observation isn't necessarily related to EVs, but it was actually going step back looking at stem cells. And the question is whether stem cell dysfunction was a consequence of aging or a cause of aging. And we have shown that if you put back young functional stem cells or young functional stem cell vesicles, that you can extend healthspan in mice. It means that loss of stem cell function, and one of that function is releasing vesicles, that if you lose that activity, that contributes to driving aging, and you can actually restore it to slow aging. So stem cell dysfunction is a cause of aging, and vesicles are one of the pathways, or release of these vesicles by stem cells is one of the ways that they function to slow aging. And the advantage to them is that unlike a protein that may bind to something on the cell surface, these vesicles can be internalized. So they can deliver DNA, they can deliver RNA, they can deliver small non-coding RNAs called microRNAs. So they can deliver things inside the cell efficiently. So there could be different types of signaling molecules than what you might view as an inflammatory cytokine or something that only binds to the cell surface. These vesicles are internalized, so they function differently than other components, other second messengers that are secreted.
I mean, if you put young vesicles into a body, then it may send signals for them.
Yes.
Yeah.
Yes, I think that the composition of or the contribution of these vesicles to all the effects we're seeing in aging is still difficult to sort out because they're very heterogeneous. All cells make them. They have a variety of components. So it's been very complicated, but we've identified some small RNAs that are carried by these stem cell vesicles that modulate senescence. So we've identified a few of these components within an extracellular vesicle that confers this anti-senescence effect, reduces inflammation. So it's probably one of the mechanisms, there are probably a lot of others, but we at least have some idea of some of the signaling mechanisms that these vesicles use to suppress senescence and inflammation.
If you use the young blood mostly from mice, it produces positive effects or youthening effects on an old rat. So do you think that is because of the exosomes in the blood? Or?
I've thought that. I've been saying that in my talk since 2014. And now a paper just came out in Nature, which I don't know why I didn't do this experiment, showing that it is indeed the exosomes from that young rat and the old rat. So that experiment was first done in the 50s. Now in Stanford and Harvard, they were recently redoing that. And there's these companies coming out of Stanford, but I'm sure I'll remember the name in a second. Ambrosia, right? The reason that you could have imagined this before, and which is why the product is actually called ExoGlow, is that when a woman is pregnant, we say her skin glows. And that's because these exosomes are coming out of the forming child into her bloodstream and circulating around and giving her that youthful glow. It doesn't end there. Multiparous women who have had many children, or animals, all live longer than the equivalent animal that hasn't had many children. So we know that the placenta's job is to make sure that cells don't get through, but exosomes can get through. So that proves that whole system that you just said there, the mice, the glowing of the woman, and why women live longer. Absolutely the exosomes.